Welcome to this tutorial about the modulation of movement by the basal ganglia. Our core concepts, as this topic relates to the field of neuroscience, again confronts the complexity of the human brain, and in particular the complexity of the motor system, and how voluntary movements are initiated through circuitry of the basal ganglia. We'll be talking about circuits that are the product of our genetic instructions, and most of the time those instructions are read out correctly and the proper circuits are constructed, but sometimes there are genetic errors that can lead to dysfunction and disability. And we'll talk about some dysfunctions of the circuitry of the basal ganglia that, at least in some cases, seem to have a genetic component to it. But thankfully, the circuitry of the brain can change. The brain is plastic, and our life experiences can change the structure and function of the nervous system. So I hope as we move on in our studies of the motor system, you'll see powerful evidence that that is in fact the case. So this tutorial on the basal ganglia is going to be broken into several parts, and I'd like to begin with part one, a discussion of the circuitry of the basal ganglia from an anatomical perspective, and then we'll move on in part two and talk about function and then dysfunction. So my learning objective for you in part one is that I want you to be able to master the anatomy of the basal ganglia, basically. I want you to identify the major components of the basal ganglia, including parts of what we'll call a dorsal motor stream and a ventral limbic stream. This word stream, as you'll see, is a word that we sometimes use in neuroanatomy to talk about a pathway or a circuit that processes information from one station to another. All right, so I'll show you again this illustration that we've seen before and use it as a way of helping you appreciate how the basal ganglia fits into our broader motor system. So you all know now that the final common path for the expression of voluntary movement are the lower motor neurons. And these exist in the spinal cord and the ventral horn and in our cranial nerve nuclei of the brainstem. These lower motor neurons are motivated by upper motor neurons. And these upper motor neurons exist in the motor cortex, in the posterior frontal lobe, and in brainstem centers, such as the reticular formation and the vestibular nuclei. So these descending systems of upper motor neurons are responsible for motivating the output of the lower motor neurons. They provide the go signal, if you will, that actually expresses the movement. They don't micromanage the movement. That's worked out at the level of our lower motor circuits in the spinal cord and brainstem, but rather they provide the command signals for the execution of that behavior. Now notice how the basal ganglia fits into all of this. The basal ganglia are off here to the side. The basal ganglia interact not directly with lower motor neurons, but they interact with upper motor neurons. So when it comes to identifying the anatomical pathways that are involved in basal ganglia function, we're going to be concerned with understanding how does the basal ganglia get information about the intention for movement, and then how does the output of the basal ganglia influence the function of these upper motor neurons. To illustrate the function of the basal ganglia, I want to bring up a component of our Sylvius anatomical software to illustrate what does it take to actually execute a movement. So what I've done is I've gone into Sylvius and I've opened up our pathways unit and selected from our animated pathways the cortical spinal tract. And what we've created here in Sylvius is just a very quick demonstration of what it takes to send a signal from our upper motor neurons in the precentral gyrus to the spinal cord for the execution of a voluntary movement. So this involves the signaling of these upper motor neurons, the propagation of an action potential, and a synaptic connection in the ventral horn of the spinal cord with lower motor neurons that then go on to express the behavior. Well, perhaps you didn't know that synapses make noise. Of course, synapses don't make noise. That's just a little feature that we put into Sylvius in order to emphasize for you that there is indeed a synaptic connection between the axons of the upper motor neuron and the lower motor neuron. All right, so I'll probably play this a few more times just because I sort of like the sound of that little synaptic connection. Okay, so the upper motor neurons send signals down the cortical spinal tract for the execution of the movement. I think we've had enough experience now with the cerebral cortex to know that if the cerebral cortex is going to generate a signal, in this case, a signal that is propagated down the cortical spinal tract, there is probably some thalamic region that is providing an important trigger signal for the processing that's happening in the cerebral cortex. And indeed, that will be the case for the motor cortex in the posterior part of the frontal lobe. There is a division of the thalamus in here that provides input to the motor cortex. You may remember from a previous tutorial, it's called the ventral anterior and ventral lateral complex of the thalamus. So this thalamic region sends signals up into the motor cortex that provide an excitatory trigger for the execution of the movement. So how do the basal ganglia factor in? Well, the basal ganglia are sitting in the deep part of the forebrain, and they provide an output to the motor thalamus, which is inhibitory. So the basal ganglia is really key for the thalamic trigger to be sent up to the motor cortex that ultimately leads in the execution of this behavior. Okay, so really the point that I'm trying to make here is that in order for the motor cortex to send out a command signal, it has to be triggered to do so by the thalamic input. And one way to control the output of the cortex is to regulate the thalamic input. And this is the function of the basal ganglia. The basal ganglia has inhibitory control over the motor thalamus. And that is a wonderful phenomenon because it keeps our movements in check. It prevents us from making all kinds of movements that we might otherwise express. So this inhibitory lock on the thalamus helps us regulate the expression of voluntary movement. Well, you may be wondering, well, how then do we move if the basal ganglia inhibits the thalamus? Well, the way we move is by temporarily removing that inhibition from the basal ganglia. And that's where the complicated circuitry comes in that links one part of the basal ganglia to another. So, as we begin to get deeper into this tutorial, you're probably wondering, well, what are the basal ganglia? I've heard that before. I remember watching some videos from the neuroanatomy lab where I pointed out the basal ganglia. But really, what are they? So let me tell you, the basal ganglia are a collection of nuclei that are found deep in the base of the forebrain. They are developed from the telencephalon, the same part of the developing nervous system that formed the cerebral cortex. Except the neurons of the basal ganglia didn't migrate out to the expanding outer sphere of the developing telencephalon. Rather, the cells that formed the basal ganglia stayed close to the base of the ventricular system, which is why the basal ganglia in the adult brain end up being very close to the lateral ventricles. Well, these basal ganglia then, they are a collection of nuclei that relate to the functions of the cerebral cortex. The basal ganglia receive input from widespread regions of the cerebral cortex. There are then several stages of processing within the circuitry of the basal ganglia, and then the output of the basal ganglia doesn't go directly back to cortex, but rather it goes to the thalamus. And then the thalamus, in turn, is what modulates the output of the cerebral cortex, in this case, the delivery of a command signal down the cortical spinal tract. Now, there are other pathways through the basal ganglia that send their output to targets in the brainstem, and I'll mention them in just a moment. But for the most part, I want you to understand that the major function of the basal ganglia is to modulate thalamocortical activity. And what I mean is the way the thalamus interacts with the cortex. In the case of the expression of voluntary movement, this is critical because the thalamus provides a trigger signal that leads typically to the expression of movement as the motor cortical circuits then send signals down the cortical spinal pathway and cortical bulbar pathway. So to control the expression of movement, it makes great sense to want to control the motor division of the thalamus. And that's what the basal ganglia do. So they play an important role in the initiation of voluntary movement, the initiation of behavior, and the suppression of behavior. That is, the suppression of movements that are inappropriate or unwanted. So keep these concepts in mind as we work through this tutorial. The basal ganglia are involved in the initiation of movement and the suppression of movement. And we'll look at the details of how that happens. Now, we've encountered this concept of parallel pathways in numerous places so far in our studies of our sensory and motor systems. So here's yet another example of parallel pathways through the brain. In the basal ganglia, we tend to call these processing streams, and what we really mean are a set of parallel pathways. And neuroanatomists have recognized actually quite a large number of processing streams that run through the basal ganglia. So we don't need to worry about all of the various ways that anatomists and physiologists have recognized parallel processing streams. Rather, we can focus on what's common about them and then recognize some key streams that we do want to focus on today. So what's common about them? Well, for each of these processing streams, there's input from the cerebral cortex to the basal ganglia. So the basal ganglia occupy these two middle layers in this schematic drawing. There's a division of the basal ganglia that we call striatum for anatomical reasons. That's what's shown here in this orange color. And then there is a division called the pallidum, also for anatomical reasons. That's shown in this purple color. So the striatum projects to the pallidum. That's fundamentally the intrinsic circuitry of the basal ganglia that we want to come to understand. And then the output from the basal ganglia is provided by the pallidum. And again, that output doesn't go directly back to the cerebral cortex. Rather, it goes back to some division of the thalamus. So, for example, our motor processing stream, sometimes we call these loops, because they begin in the cortex and they ultimately come back to impact activity in the cortex. So our loop for body movement is derived from cortical inputs that converge from a widespread region of the cerebral cortex into the basal ganglia. So here's the cortical input coming into a part of the basal ganglia that we call the putamen. And that division, part of the striatum, then sends signals into the globus pallidus, another part of the basal ganglia that's representative of this layer of tissue and of processing that we call the pallidum. The globus pallidus, in turn, projects to the motor thalamus, this ventral anterior ventral lateral complex. And the motor thalamus finally sends signals into the motor cortex that can either trigger the initiation of movement or, depending upon the physiology of the basal ganglia, suppress unwanted movements. So this same kind of theme of cortex to striatum, striatum to pallidum, pallidum to thalamus, and then thalamus back up to cortex is repeated in each of these processing streams. And we have something of a simplified view in front of us where we simply recognize that there are motor loops or motor processing streams and non-motor loops. The motor loops concern movements of the body, and there seems to be a dedicated processing stream for movements of the eyes. And on the non-motor side, we can recognize a loop that is concerned with the functions of the prefrontal cortex that we'll talk about in a few tutorials. This plays an executive functioning role in cognition. And there are processing streams that seem to involve the limbic parts of the forebrain. And these are brain regions that are involved in the experience and expression of emotion. So the loops are primarily defined by the anatomical inputs from the cerebral cortex and the kinds of functions that are carried out by those cortical areas that receive the feedback that comes from the thalamus after being modulated by the basal ganglia. So again, I would encourage you by saying don't worry about the complexity of this figure. Just understand the point that there are parallel processing streams that are concerned with the initiation and suppression of movement in one domain of physical action or mental action or emotional action or another. Well, one important means by which we've come to understand the functions of the basal ganglia in the normal human brain has been based on studies of individuals that have had disease or disorder in the circuitry of the basal ganglia. And so by understanding a neurobiological framework of disease in the basal ganglia, we've gained considerable insight into the normal functions of these parts of the brain. So we will take some time in this tutorial to consider two major classes of movement disorder that relate to basal ganglia function. As you'll come to see, we will recognize a category of movement disorder where the problem is not enough movement. We call these hypokinetic movement disorders. And the chief representative in this category would be Parkinsonism. And we'll come to understand Parkinsonism as primarily a failure to initiate voluntary movement. Of course, there is much more to this clinical picture than just this, but for our purposes, focusing on the role of the basal ganglia, Parkinsonism is a failure of initiation. On the other end of the spectrum, there are hyperkinetic movement disorders. So these are disorders that are represented by Huntington disease and by other kinds of disorders that result in the expression of unwanted movement. So we can think of these disorders in terms of the basal ganglia as a failure to suppress unwanted movement. Now, what's been very exciting in recent years is the possibility that this concept of hypokinetic disorder and hyperkinetic disorder might also apply to some of these non-motor loops that are running through the basal ganglia. Perhaps various affective and cognitive disorders reflect either a failure to initiate a particular behavioral program or a failure to suppress an inappropriate behavioral program. So this is an exciting hypothesis that is getting much attention and research by cognitive neuroscientists and psychiatrists. Okay, so this has really been an extended introduction to the components of the basal ganglia and a little bit about their function. Now let's turn to a sharper focus on the major parts of the basal ganglia. And we're going to focus on really just two of these processing streams, two of these loops. I'd like for us to consider the organization of the body movement loop and the organization of the limbic loop. So the body movement loop is a circuit that runs through the basal ganglia that's primarily concerned with the movements of our limbs. The limbic loop is a circuit that runs through the basal ganglia that's concerned with our affect and our motivation. And that will be important when we consider the neurobiology of addiction. All right, so let's look at what's common about these two parallel processing streams that run in the basal ganglia. They both have cortical input. In the body movement loop, the cortical input is primarily from the motor cortex and the parietal cortex, where we find our somatic sensory signals. So the input is coming from a fairly large region of the frontal parietal area of the cerebral mantle. In the limbic loop, the cortical input is coming from telencephalic structures that we associate with the ventral and medial parts of the forebrain. This includes the amygdala, the hippocampus, the orbital and medial parts of the frontal lobe just in front of the motor cortex. This includes the cingulate gyrus and regions of the temporal cortex. This cortical input comes into striatum, but to a region that we call the ventral striatum, which, as we'll see in just a moment, sits inferior to our putamen. And there's a division of this ventral striatum that I'm going to want to highlight for you that's called the nucleus accumbens. And I'll show you that region when we look at some sections through the basal ganglia. It's worth mentioning now because the nucleus accumbens is ground zero for research on addiction in the human brain. Well, The striatum then sends inputs to the pallidum, and for the body movement loop, the pallidum is the globus pallidus, in particular the internal segment of the globus pallidus. For the limbic loop, the pallidal region is called simply the ventral pallidum. It's a division of the pallidum that's inferior and just a little bit posterior to that part that we recognize as the globus pallidus. Now, the pallidum provides the output from the basal ganglia. So in the body movement loop, the pallidum here is the internal segment of the globus pallidus, which projects to our motor thalamus. The motor thalamus, in turn, sends signals back into the motor cortex. In the limbic loop, the ventral pallidum projects to a different thalamic structure. It's called the medial dorsal nucleus of the thalamus. And this is the thalamic nucleus that sends signals back to many of these telencephalic structures that provided the original input to the ventral part of the striatum. In particular, the cingulate gyrus and other parts of the orbital and medial prefrontal cortex. So again, the point here is to lay out the specific processing stations that are involved in these different processing streams, but also to highlight what's common here: cortex to striatum, striatum to pallidum, pallidum to thalamus, and then thalamus back to cortex. Okay, I think we're ready to actually look into the brain and see what some of these structures actually are. So first, with an illustration, what we have here is a coronal section through the forebrain, and this allows us to see the various components of the striatum that are visible at this level. We see the putamen and the caudate nucleus, divisions of the striatum that are separated by the internal capsule. Just on the medial side of the putamen is where we find the globus pallidus, and as we'll see, the globus pallidus has two parts to it, an external part and an internal part. The output from the pallidum is directed towards the thalamus, and we see our motor thalamus here in this level. This is a structure that we recognize as the ventral anterior ventrolateral complex of the thalamus, and those terms provide some indication as to what part of the thalamus is in question. And then, of course, that part of the thalamus is what gives rise to projections on up to the motor cortex. Now, this circuitry is modulated in some very important ways that we're going to spend some time talking about. The principal modulation that we will consider is coming from the midbrain, and it comes from a division of a region that we call the substantia nigra. Now, you may have heard about the substantia nigra. You may have seen the substantia nigra. Substantia nigra is one of those wonderful terms in brain anatomy that tells you what it is. It's the black substance. If you look at a brain at autopsy and make a section through the midbrain, you will see a dark substance that forms a bit of a line that is nearly sitting on top of the cerebral peduncles. So that dark substance is what we call the substantia nigra pars compacta. The dark substance is there as a result of the biosynthesis of the neurotransmitter that's released by these cells. That transmitter is dopamine. And over a lifetime of making dopamine, these cells accumulate a dark pigment, which gives the dark substance its name. Dopamine is released at the axon terminals of these cells, and the projections from this pars compacta of the substantia nigra is targeted to the striatum, to the putamen, and to the caudate nucleus. So dopamine from the midbrain becomes an important neuromodulator of basal ganglia function. We'll spend a bit of time talking about that. There's another structure that provides an important modulatory influence over the basal ganglia, and that is the subthalamic nuclei. The subthalamic nuclei sit basically just in front of the substantia nigra, on top of the cerebral peduncles, at a junction between the diencephalon and the midbrain. But the subthalamic nuclei are part of the diencephalon. The subthalamic nuclei provide input to the internal part of the globus pallidus and help to sustain activity that drives the output from the internal segment of the globus pallidus to the thalamus. So one might quibble about whether the subthalamic nucleus are really part of the basal ganglia. Because they're diencephalic, I don't include them as part of the basal ganglia directly. Rather, I consider this to be an important source of modulation of basal ganglia function. Now, it's not illustrated here, but I'll also point out, while we have the substantia nigra in front of us, that there is a medial region in the vicinity of the pars compacta that likewise contains a collection of cells that make dark substance. This is what we refer to as the ventral tegmental area, or the VTA for short. The ventral tegmental area provides a dopamine projection to the ventral part of the striatum that participates in the limbic processing stream. All right, well, let's look now at the forebrain and try to appreciate the complex geometry of the basal ganglia. And I'll start actually with our brain model that we have here. So I've alluded a few times, as our focus has been on the brain stem, that there are structures that are part of the forebrain that are sitting on top of this brain stem in this model. And what we're looking at basically is the basal ganglia. So from the frontal view, we're looking at the putamen laterally, and on the medial side is the caudate nucleus, parts of the striatum. And where the caudate nucleus and the putamen join together down below, that's the region that we call the ventral striatum. And specifically, there is a division here that we call the nucleus accumbens. So the nucleus accumbens is simply the inferior and anterior continuity of the putamen and the caudate nucleus. Now, I want to show you the posterior view, which we would only see if we dissected away the forebrain. And let me first orient you a little bit. So this bundle of white matter that we find right here, this is called the internal capsule. And it's an important landmark that will help you recognize what parts of the basal ganglia you're looking at. And what you should remember is that the putamen is on the lateral side of the internal capsule, whereas the caudate nucleus is on the medial side of the internal capsule. Now the putamen and the caudate, as I just mentioned, they join together around the anterior side of the internal capsule with the nucleus accumbens, forming the inferior continuity. But on the posterior and dorsal aspect of the basal ganglia, the caudate nucleus extends a long tail that runs along the medial edge of the internal capsule. And it actually extends all the way down into the temporal lobe, which is not illustrated here in this model. But we'll see a long tail of the caudate that extends quite a distance into the temporal lobe following the course of the lateral ventricle. Now, there's an important basal ganglia component that we cannot see in this model, and that is the globus pallidus. The reason that we can't see it is that it's buried just on the medial edge of this putamen. It sits between the fibers of the internal capsule and the putamen. Now, if we look at this illustration, hopefully you'll have some orientation now as to what we're seeing. So from this lateral view, we can appreciate the putamen, that division of the striatum that we find most laterally. And the putamen is continuous in its anterior extent with what we call the head of the caudate nucleus. And now from this illustration, you can see how the caudate nucleus wraps all the way around the body of the lateral ventricle and follows the temporal horn of the lateral ventricle into the temporal lobe. Now again, what we don't see here in this view is the globus pallidus, because the globus pallidus is completely enveloped by the putamen. But what we do also appreciate is the position of the thalamus. So here's the thalamus. That is a large egg-shaped structure that's on the medial side of the internal capsule, and it is a little bit more posterior in its extent than is the putamen. So we'll note that the thalamus seems to be shifted just a little bit in the posterior direction as we look at cross-sections through the brain. So let's go ahead and do that now. All right, so we are looking at a coronal section through the forebrain, taking at a level that is just catching the anterior tip of the temporal lobe. And I want you to notice, of course, all the cortex that we find forming the bark. Around the margins of the cerebral cortex, we have white matter underneath. But now we've got this deep gray matter that is bulging out into the lateral wall of the lateral ventricle, and it seems to be surrounded superiorly, laterally, and inferior with the white matter of the hemisphere. So let's look at what we have here. We are looking at a section that would have been taken very near the front edge of the internal capsule. So what we would expect to see is this continuity of the caudate, the putamen, and then the ventral striatum, the nucleus accumbens down below. And that's really what we observe here. So you'll notice just a little wispy bit of white matter that is labeled number 6 here in the left hemisphere. That's the front edge of the internal capsule. So that must mean then that what we have in this view is all striatum. So we have the caudate nucleus on the medial side of the internal capsule, the putamen on the lateral side, and where the two come together, we have the nucleus accumbens. Well, I'd like to make just one additional point before leaving this section to move on into the basal ganglia. And what I'd like to talk about are these parallel streams that run through different divisions of the striatum. So again, what we have in view here are the different parts of the striatum. Out here laterally, we have the putamen, which is on the lateral aspect of the internal capsule. On the medial aspect of the internal capsule and bulging in to the lateral wall of the lateral ventricle, we have the caudate nucleus. And then where the caudate nucleus and the putamen join together, inferior to the anterior limb of the internal capsule, that's what we call the ventral striatum, or in this region, primarily we call this the nucleus accumbens. Now, I'd like to emphasize that the basal ganglia is all about modulating movement. And what distinguishes one stream from another is what is the object of the movement. Now, I'm going to ask you to give me a little bit of license with this idea of movement and apply it somewhat creatively. Well, let's begin with a more concrete application of the concept of movement. The putamen is all about moving the body. So the putamen is the principal recipient of the inputs from the frontal motor cortex and the parietal somatic sensory cortex. So the putamen is about modulating the movements of the body. The caudate nucleus receives input from the frontal cortex, specifically the parts of the prefrontal cortex that sit just in front of the motor cortex. The caudate nucleus is also receiving input from the anterior part of the premotor cortex, which would include our frontal eye field. So the caudate nucleus is concerned with movement of the eyes, and with the eyes also goes our mind, our cognitive faculty. So I like to think of the caudate nucleus as mainly modulating movement of mind, that is, going from one thought to another, often as we visually inspect our world. So what we look at is typically what we're thinking about. Not always, but for the most part, eye movements and thought go together. So the caudate is moving the mind. The nucleus accumbens is receiving input from the ventral medial parts of the forebrain that are concerned with processing emotion or affect. So the nucleus accumbens is going to be involved with moving affect. So what do I mean by moving affect? Well, I mean the nuances of our emotional life as we go from one moment in our day to another. We have ups and downs in our emotions, and that's normal. We ought to have a normal emotional response to the events in our lives that move our emotions. That movement of emotion is facilitated by the circuitry that run through the ventral part of the basal ganglia, including those circuits that are converging in the nucleus accumbens. So please remember, the basal ganglia modulate movement, and they do so operating upon different modalities of information that are converging on different parts of the striatum. Okay, let's move on to the next section now. If we go back a section, now we are cutting through really the fat part of the internal capsule. So what we have again is this now broad internal capsule that helps to orient us to our view, but we still have elements of the striatum that we saw in the previous section. The caudate nucleus is bulging out, forming the lateral wall of the lateral ventricle. And that's a nice, healthy-looking caudate nucleus. That's what we should see. And then now we have the putamen, which is lateral to the white matter of the internal capsule and inferior to the location of the caudate nucleus. But there is now the globus pallidus taking shape between the fibers of the internal capsule, the putamen, and this other white matter pathway that we see now extending across the midline. So perhaps some of you know what this white matter pathway is down here. I'll give you just a moment to shout it out. Hopefully you will recognize this as the anterior commissure. So the anterior commissure is a helpful landmark because it helps us recognize where we would expect to find the globus pallidus. The globus pallidus sits in a bit of a triangular region between the putamen, the internal capsule, and the anterior commissure. In the next section, we are getting near the posterior aspect of the basal ganglia. So perhaps we have a section that is roughly in this orientation here. So we are losing the putamen and the globus pallidus. It's still present, as is our caudate nucleus. So again, let's reorient. Here is our internal capsule that's streaming into the region of the cerebral peduncle. And on the medial side of that capsule, we expect to find the caudate nucleus, which is this little bit of gray matter here. It's getting smaller because now we're developing that tail that's going to follow the course of the lateral ventricle into the temporal lobe. On the lateral side of this internal capsule, we have the putamen. It's smaller than it was in the previous image, but it's still present. And we also have just a small bit of our globus pallidus left. It is just where we would expect it to be. It's between the internal capsule and the putamen. Another major component that's made its appearance in this section is, of course, the thalamus, which is this large egg-like structure that we see on the medial side of the internal capsule. Now, you may be wondering about this limbic loop, where are the structures that the nucleus accumbens projects to, and so on. So let me just point those out while we're making our way through these sections. So I did, in fact, show you the nucleus accumbens, which is this ventral and anterior region where the caudate nucleus and the putamen seem to fuse together around the anterior margin of the internal capsule. The nucleus accumbens sends inputs into the ventral pallidum, and I recognize this triangular region as the globus pallidus. The ventral pallidum begins to take shape just underneath this anterior commissure and extends posteriorly just a little bit. So this is the region that we would call the ventral pallidum. And the ventral pallidum provides the output from that limbic loop to the thalamus. And specifically, that output is directed to the medial dorsal nucleus of the thalamus, which is present right about there. So there's our medial dorsal nucleus. The medial dorsal nucleus provides output to the cingulate gyrus and other divisions of the prefrontal cortex. Okay, so now that you've seen the basic parts of the basal ganglia, let's begin to consider the connections among the various parts. So I've already talked a little bit about this first point. What are the major inputs to the basal ganglia? Hopefully you will recall that the inputs to the basal ganglia are directed towards the striatum. So much of that input is derived from the telencephalon, from the cortex. So we see numerous projections from the cerebral cortex into the striatum, which is the caudate nucleus and the putamen. So all of this cortical input's converging at the level of the striatum. The striatum is also the target of our dopamine inputs from the substantia nigra pars compacta. Okay, so the dopamine is being released in the striatum. That's going to be very critical for understanding how the circuitry of the basal ganglia function. So the striatum is really the key input station for all of the loops that run through the basal ganglia. For the limbic loop in the basal ganglia, same principles at play. We have projections from cortex, only for the nucleus accumbens, the cortex that's providing input would be in the temporal lobe, as well as in the orbital and medial parts of the prefrontal cortex. So these inputs from structures like the amygdala, parts of the hippocampus, and then that orbital medial prefrontal cortex are converging on the striatum. And in this case, it's the nucleus accumbens. Our dopamine cells for the nucleus accumbens, again, are derived from the medial part of the dark substance. That's what we call the ventral tegmental area. And just as for the dorsal processing streams concerned with moving the body, so in this limbic loop, dopamine is released in the striatum, in the nucleus accumbens. This will be very important for understanding our neurobiological framework for addiction. I think we're now ready to talk about the circuitry within the basal ganglia now that we understand the convergence of inputs into the striatum. So we conventionally talk about two pathways that get information through the circuitry of the basal ganglia and into the thalamus. There's a direct pathway because it's the most direct connection from the striatum to the pallidum and then out to the thalamus. And here's the anatomy of the direct pathway through the basal ganglia. We've already talked about the inputs from the cortex that come into the striatum. So collectively, we can collapse the striatum and call it the caudate/putamen. So this is just the striatum. The striatum sends projections to the pallidum. In the case of our body movement loop, that would be the internal segment of the globus pallidus. The internal segment of the globus pallidus then sends signals to the motor thalamus, the ventral anterior and ventral lateral complex of the thalamus, which in turn provides input to the motor cortex. Now don't worry about the pluses and the minuses and the transients and the tonics at this point. We'll come back to that. For now, I want you to focus on the anatomy. So again, we have cortex projecting to striatum, striatum to pallidum, pallidum to thalamus, and then thalamus back to cortex. This is the direct pathway. There's an indirect pathway. The indirect pathway actually, I must admit, looks worse than it is. What we have in this illustration is a combination of the direct and the indirect pathway. So the direct pathway is what we find to the right-hand side of this figure. The indirect pathway is the portion of this figure that is backed by that yellow field. So the indirect pathway, as its name suggests, must be a less direct means of getting information from the striatum through the pallidum and onto the thalamus, and indeed it is. The indirect pathway involves an additional loop that runs from the external segment of the globus pallidus through the subthalamic nucleus and then back to the internal segment of the globus pallidus. So let's see how that works out. So here's our indirect pathway now. This area that's backed by this yellow color. And our indirect pathway involves projections from cortex to the striatum, projections from the striatum now specifically to the external segment of the globus pallidus. So this is the more lateral division of that globus pallidus body that we saw a moment ago. The external segment of the globus pallidus projects to the subthalamic nucleus, and it also has direct projections to the internal segment. It seems as though this projection to the subthalamic nucleus is really important in how the indirect pathway works, because the subthalamic nucleus then projects back to the internal segment of the globus pallidus. And now we're back into the confluence of the direct and the indirect pathways. The internal segment of the globus pallidus projects to the motor thalamus, which then sends signals into the motor cortex for the initiation of movement. So when one considers the indirect pathway, what I want you to appreciate is the importance of this subthalamic nucleus. If the function of the basal ganglia is to modulate the way the thalamus is interacting with the cortex, then you might imagine that the subthalamic nucleus ought to be pretty important because it's one way to either turn up or turn down the activity of this internal segment of the globus pallidus. Well, so is the direct pathway for that matter. But these two inputs converging in the internal segment of the globus pallidus are very important in modulating or controlling the output of the basal ganglia that ultimately is reaching the thalamocortical circuit. So after a short break, let's come back and let's consider the function of this circuitry in the basal ganglia. Okay, in part 2 of this tutorial, what I want us to do is to consider the function of the circuitry in the basal ganglia. So my learning objectives for you are that I want you to be able to discuss the role of the basal ganglia in the initiation and suppression of behavior. That's ultimately what this circuitry is going to accomplish. I want you to understand the important principle of disinhibition, which is played out all over the nervous system, but here in the basal ganglia it's especially critical for understanding how this circuitry works. So I want you to be able to explain how this concept of disinhibition applies to the circuitry and to the functions of the basal ganglia. And I want you to be able to discuss the critical role of dopamine in facilitating the function of these basal ganglia circuits, because as I hope to impress upon you, dopamine really makes the basal ganglia function. It facilitates the balance of activity that's essential for the role of the basal ganglia in the modulation of movement. So let's consider this notion of disinhibition. So what is disinhibition? Disinhibition is the inhibition of inhibition. Did you follow that? So the implication here is that there are two inhibitory neurons connected to one another in series. So I really want you to study this figure that we have in front of us. You can either look at it in your textbook or just freeze frame this video and make sure you understand conceptually what's happening. So again, for disinhibition, we need to have two inhibitory neurons connected up in series, and that's what we have here. So neuron A is releasing an inhibitory transmitter on neuron B. Neuron B is releasing an inhibitory transmitter on neuron C. So these are our two inhibitory neurons. Inhibitory neuron number 1 and then inhibitory neuron number 2. That provides us with the basic ingredients for disinhibition. So while this figure is largely conceptual and highly schematic, it's intended to represent the actual major cell types that are found in the basal ganglia. So notice, for example, that the striatal neuron, represented by this neuron A, is receiving inputs from the cerebral cortex that are driving the activity of the striatal neuron when these cortical inputs converge in space and time in the striatum. Now, that excitatory drive is going to lead to the release of inhibitory neurotransmitter on the globus pallidus. Since the globus pallidus itself is releasing an inhibitory neurotransmitter on the thalamic neuron, then activity in the striatum is going to shut down the activity of the globus pallidus. And if we remove the inhibition of the globus pallidus on the thalamus, well, I hope you'll understand that that means that this thalamic neuron is going to release neurotransmitter and drive activity in the motor cortex that then can lead to the generation of action potentials down the cortical spinal tract. So I'll give you just another moment to look at this figure and consider how this works. And again, the key feature here is to recognize that if the functional goal is to trigger activity in the motor cortex, what is necessary then is to remove the inhibition on the thalamic projection to the cortex. When we remove that inhibition, that releases the motor division of the thalamus to fire action potentials and trigger the activation of our cortical spinal pathway. The circuitry of the basal ganglia then, in the direct pathway in particular, is set up to remove that inhibitory lock that the internal segment of the globus pallidus has on the thalamus. So let's look at this now with a little bit of schematic physiology there to guide our understanding. So imagine now that neuron A in the striatum is at rest. So there's not a particular plan to move, a desire to move, an urge to move. The motor cortex is essentially at rest. So the relevant part of the striatum is going to be silent. Well, for reasons that I haven't mentioned yet, the activity of the internal segment of the globus pallidus is quite high. So there is tonic activity being generated here in this globus pallidus that is causing inhibitory neurotransmitters to suppress the firing of the ventral anterior and ventral lateral nucleus of the thalamus. So when we have all this tonic activity in the internal segment of the globus pallidus, we can expect there to be profound inhibition in the VA-VL complex. And without activity coming out of the motor thalamus, we would expect little activity, if any, being generated by the cortical spinal tract neurons of the motor cortex. So that's the situation at rest. Now, what about when we have a desire to move, when there's a convergence of inputs from the cortex into the striatum, and that's going to transiently drive the activity of this striatal neuron. And so that striatal neuron then is going to be releasing its inhibitory neurotransmitter in the globus pallidus. So when the striatal neuron is excited, we expect there to be inhibition at the same moment in time of the internal segment of the globus pallidus. If we remove the inhibitory lock of the globus pallidus on the thalamus, then the thalamus is free to have a release from that inhibition. So for this reason, we say that the thalamus is what has been disinhibited. So the inhibition of inhibition has released the thalamus to fire activity, which then drives the output of the cortical spinal neurons. So one question I often like to ask is, what gets disinhibited with the activation of this direct pathway? The answer is that it's the thalamus that gets disinhibited. And the logic there is that the thalamus is at the end of this two-neuron chain that puts two inhibitory neurons back to back. And that's the circuitry of the direct pathway. Okay, now let's put together the direct and the indirect pathway and see how they function. The direct pathway, as I just illustrated for you, facilitates the activation of the frontal motor cortex. The indirect pathway works to suppress the activation of the motor thalamus and therefore the output of the motor cortex. So the indirect pathway then would reinforce the tonic inhibition that clamps down the activity of the motor thalamus. And here's how this works. So let's focus now on the indirect pathway. So the indirect pathway again is here on the left side of the figure. We imagine that there are some striatal cells that are being driven to fire action potentials by input from the cortex. Those striatal cells send inhibitory projections to the external segment of the globus pallidus. This means that the tonic inhibition of these cells in the external globus pallidus is transiently suppressed. Well, when that happens, then we expect there to be an increase in activity in the subthalamic nucleus. So in the indirect pathway, it's the subthalamic nucleus that is disinhibited. That will drive activity, releasing excitatory neurotransmitter in the internal segment of the globus pallidus. So what would happen if we increased the output of the internal segment? Well, that's going to drive up the tonic inhibition of the internal segment on the motor thalamus, making it very unlikely that the motor thalamus would send a trigger signal to the motor cortex to release movement. There's also this direct projection from the external segment of the globus pallidus to the internal segment. And, well, if we suppress the activity of that external segment, then we will see a reduction in the release of inhibitory transmitter. That too will synergize with the disinhibition of the subthalamic nucleus. These two connections put together leads to an increase in the inhibitory output of the internal segment of the globus pallidus. So you can see how the connections through the internal segment work to suppress movement. They work in a push-pull fashion with the direct pathway that converges on the internal segment of the globus pallidus, with the direct pathway facilitating the expression of movement and the indirect pathway suppressing movement. Now, if you're following all this, you should be wondering at this point, well, how is it that we ever make movement if these two parallel streams through the circuitry of the basal ganglia seem to be fighting each other? Well, that's where dopamine comes in. So let me just highlight the importance of dopamine for the function of the basal ganglia. And something really interesting is happening here. Dopamine is a biogenic amine neurotransmitter, which is synthesized and released in the striatum, and it binds to two different types of receptors. They're both metabotropic receptors, but they link to second messenger systems that produce opposite effects on the level of cyclic AMP within the postsynaptic neurons. The D1 receptors lead to an elevation in cyclic AMP. So when D1 is activated, cyclic AMP levels rise, and that has a facilitatory effect on the postsynaptic neuron. When the D2 receptors are bound by dopamine, that leads to a reduction in cyclic AMP levels. And that leads to a suppression of neuronal activity in the postsynaptic cell. So dopamine is a key regulator of the responsiveness of the postsynaptic neuron to the input that it receives from the cortex. And that postsynaptic neuron is going to be a medium spiny neuron of the striatum. And here's what's important to understand about this. The D1 receptors are expressed by neurons of the striatum that are in the direct pathway from the striatum to the internal segment of the globus pallidus. The D2 receptors are expressed by striatal neurons that are in the indirect pathway from the striatum indirectly to the internal segment of the globus pallidus. So when dopamine is released in the striatum, here's what happens. The activity of the direct pathway striatal neurons goes up, and the activity of the indirect pathway striatal neurons go down. And when we activate the striatal neurons of the direct pathway, movement is facilitated. When we suppress the activity of the indirect pathway, it synergizes with the activity of the direct pathway. So dopamine turns this tug of war between the direct and the indirect pathway into a synergistic function where both of these circuits are now working together. So it's so important for you to settle in on this figure and understand why dopamine makes this all work. Well, it may be difficult for you to do that with the complexity of this figure. Let me give you a suggestion on how to approach your studies. Why don't you start with the idea of what does it take to move the body? Well, the cortical spinal neurons of the motor cortex have to engage our lower motor neurons. Well, start there and then back into this complicated figure, and I think it will be much more easy for you to understand. So let's try that. Well, in order to activate our cortical spinal neurons, there has to be a trigger signal from the thalamus. Well, how does that happen? We have to disinhibit the thalamus because, remember, it's at rest clamped down by an inhibitory output from the basal ganglia. So if we have any hope of expressing movement, then we need to take out the tonic inhibition from the internal segment of the globus pallidus. And that's where the direct pathway comes in. We inhibit the inhibition, and that leads to a reduction in the suppression of the thalamus, which causes a release or a disinhibition of the VAVL complex and activation of our cortical spinal neurons. The indirect pathway will synergize beautifully in the presence of dopamine, because dopamine is serving to increase the activity of the direct pathway by binding to a D1 receptor, but dopamine is suppressing the activity of the indirect pathway by binding to D2 receptors. So if we suppress the activity of the indirect pathway, that's going to lead to an increase in this inhibitory output of the external segment of the globus pallidus. That will contribute to the shutting down of the internal segment of the globus pallidus, which will facilitate movement. So the internal segment is being suppressed by this direct projection, and the excitatory drive on the internal segment from the subthalamic nucleus is being shut down. So you see, in the presence of dopamine, the direct and the indirect pathway become synergistic partners for the facilitation of movement. And we'll talk about in just a few minutes the impact of Parkinson's disease, which involves a loss of dopamine. And I think now you should appreciate how important dopamine is for the synergistic function of this circuitry of the basal ganglia. And hopefully that gives you some insight into the problems that we would face if we didn't have enough dopamine to shift the balance and activity in favor of the direct pathway and the expression of movement. Now, what I've been illustrating for you is the circuitry involved in the body movement loop, or the dorsal processing stream through the basal ganglia. Something quite similar is happening for the ventral stream. There are direct and indirect pathways that are running through these ventral parts of the basal ganglia. I won't show that for you here, but I do want you to know that it's occurring in exactly the same way as we see for the body movement loop. It's just running through different parts of the basal ganglia, through the ventral striatum and the ventral pallidum, and then out to a different thalamic nucleus, which relates back to a different division of the cortex, the prefrontal cortex. Okay, we have come to our next pause, and so let's go ahead and take a short break, and then please do come back, and we'll pick up with the third part of this tutorial, where we'll talk about normal and abnormal modulation of movement by the basal ganglia. Welcome back to this tutorial on the modulation of movement by the basal ganglia. And now we turn to part 3, which is about normal and abnormal modulation of movement by the basal ganglia. So my learning objectives for you are, first, that I want you to explain the hypokinetic movement disorders in terms of the function of basal ganglia circuitry, and I want you to consider the hyperkinetic movement disorders in terms of the function of this same circuitry. So to set the stage for this discussion of abnormal function, let's first recap what we've understood about normal basal ganglia function. So the neurons of the striatum, they begin to fire just before and during the onset of movement. So this suggests that the role of the basal ganglia is the initiation and the suppression of movement. But the basal ganglia are much less critical during the ongoing expressions of a motor program. As we'll see in a future session, that's the function of the cerebellum. And what I want you to understand, that it's the balance in the activity between the direct and the indirect pathways that mediates the initiation and the suppression of movement. By analogy, we can think about this as a kind of center-surround antagonism, not for receptive field structure, but relative to what we can possibly do with our motor system. Let me illustrate what I mean with a highly conceptual figure. let's imagine that we intend to make a voluntary movement. So this is where we want to be. We want to activate a specific motor program that's appropriate for the movement goal at hand. Well, in order to do that, what must happen is we need to release the activity of the motor thalamus, which can drive the output of the cortical spinal neurons in the motor cortex. Release of the motor thalamus means that we need to have a way of shutting down the inhibition of the internal segment of the globus pallidus. And that's where this direct pathway comes in. So the direct pathway, via the caudate and the putamen, sends inhibitory signals into the internal segment of the globus pallidus, which then removes the inhibition. Consequently, the motor thalamus is released and fires action potentials that then trigger the output of the motor cortex. So this is what happens with respect to the motor programs that we actually intend to activate. Well, what I want you to appreciate is that there is a large surround of possible movements that could interfere or antagonize the desired motor program. So while we have a specific program in mind to activate, we also want to enforce the suppression of all other possible motor programs that pertain to the same musculoskeletal units. And this is where this concept of surround inhibition, I think, might be useful. So we can think of this surrounding repertoire of movement that we want to suppress as being under the influence of the indirect pathway. And that surround comes in play schematically by this annulus. So we imagine that there is a kind of movement surround being represented in the thalamus that we want to suppress with this activation of the indirect pathway. So if anything, to facilitate the specific expression of movement, we want to make sure that non-synergistic movements or antagonistic movements are held in check. So the activity in the indirect pathway then serves to reinforce this inhibition of this surround of potential movement. And that's where the subthalamic nucleus comes in. So when we activate the indirect pathway, we are driving the subthalamic nucleus to increase the inhibitory output of this internal segment of the globus pallidus. So this center surround model, I think, is a useful way of considering how is it that we can execute a very particular movement, like a visually guided reach. So as I'm expressing this sort of reaching and pinching motion, I'm suppressing all other kinds of activities that potentially could be done with the same musculoskeletal units. And so the focused activation of a very particular motor program requires a shift in balance in favor of the direct pathway for this particular motion and the suppression of all the other unwanted movement programs that potentially could interfere with the goal of the behavior. Now, I want to apply some of these concepts to some of the disorders of movement that you'll see clinically. And as I've suggested in our opening, there's really a continuum of movement disorders. On one end of the continuum, we have disorders that produce too little movement, and on the other end of the continuum are disorders that allow too much movement. So I want to begin first on the hypokinetic side of that continuum and use a rather familiar example, unfortunately, and that's Parkinson's disease or Parkinsonism, which refers more to the symptoms associated with idiopathic Parkinson's disease. So those symptoms involve a reduction in movement or perhaps even a cessation of movement. We call this akinesia or bradykinesia. It involves a highly rigid posture. That rigidity is not exactly the same thing as the spasticity that's seen with damage to upper motor neurons. Rather, it reflects a suppression of movement that is reinforced by the overactivation of the indirect pathway. Somewhat paradoxically, there is a resting tremor that's seen in Parkinson's disease, particularly in the hands. Sometimes this is called a pill-rolling tremor, as if the hands of the patient are trying to roll a pill in the fingertips. But sometimes the tremor can be a little bit more proximal and can be expressed at rest. So it seems somewhat paradoxical that we might have a slowness of movement or an absence of movement together with a positive sign like a tremor, but it reflects the activity that is disordered in different parts of the basal ganglia. Well, the bradykinesia doesn't only affect the appendicular movements that we do with our limbs, but it also affects our facial expressions. So people who are afflicted with Parkinson's disease, they tend to have a reduction in facial expressions. So with the problems attending bradykinesia, it's not surprising that there would be disorders of gait. And there's a very characteristic gait pattern that's seen in people with Parkinsonism. It is a shuffling of the feet with very small steps and very little elevation of the foot above the walking surface. So one of the characteristic signs of Parkinsonism is difficulty initiating and terminating movements. So it's very, very difficult to get a motor program started, and then once that program is started, it's difficult to terminate it precisely. So we can think of this as a real challenge in shifting from one motor program to another, from go to stop and to go again. Eventually, there are changes that afflict not just the motor system, but other parts of the brain as well. So quite tragically, advanced Parkinson's disease often leads to the development of cognitive decline and dementia. Now, in idiopathic Parkinson's disease, we typically see these symptoms setting on after the 5th decade of life. So the underlying neuropathology that makes it appropriate to consider this disorder here in this discussion of the basal ganglia is that in Parkinson's disease, there is a loss of these dopamine neurons that are so important for regulating the balance between direct and indirect pathways. So in order for a patient to present clinically, they need to lose some 75 to 80% of their dopaminergic neurons. So there is a bit of redundancy built into this dopaminergic system, thankfully. But nevertheless, with loss of these dopamine neurons beyond that critical threshold, one can see the development of the signs and symptoms that I just described for you. So let's look at the circuitry of the basal ganglia and consider the consequences of losing this input from the substantia nigra pars compacta. What we would find is the inability to facilitate the activity of the direct pathway and the inability to suppress the activity of the indirect pathway. These two factors synergize in a rather unfortunate way that results in an increase in the tonic output of inhibition from the internal segment of the globus pallidus to the motor thalamus. So the problem in Parkinsonism is bradykinesia, a slowness of movement, an absence of movement. That's because the motor thalamus is tonically suppressed, and it's very difficult to overcome that suppression. As a consequence, there's decreased excitation of the motor cortex and tremendous difficulty initiating movement. So now let's back up into the basal ganglia and see why that would be. So if there's too much tonic inhibition, that suggests that the activity in the internal segment of the globus pallidus is too high. And again, the reason why it's too high is that we have a severe reduction in our ability to inhibit the inhibition. So we have a failure of disinhibition. Matters are made worse by the absence of dopamine on our D2 receptors, which means that we cannot suppress the activity of this indirect pathway. So we have a lot of inhibition coming out of the indirect pathway, which will shut down the external segment of the globus pallidus. And that means that it's likely that we will have disinhibition of the subthalamic nucleus. So the subthalamic nucleus is the main culprit here, because it's driving the increased tonic outflow from the internal segment of the globus pallidus. Well, really the principal culprit is the loss of these dopaminergic neurons, but we can see the importance now of the subthalamic nucleus in modulating the output of the basal ganglia, because if we cannot reduce the tone of the subthalamic nucleus, we will see a marked elevation in the inhibitory output of the internal segment onto the thalamus. The end result of all of this is bradykinesia, perhaps even akinesia. Well, thankfully, collaborations between basic scientists, be they neuroscientists or biomedical engineers, and the clinical communities have led to the development of new interventions. One that I would invite you to read more about is deep brain stimulation. Now, the idea with deep brain stimulation is to stimulate strategic locations in the circuitry of the brain that will produce a desired clinical outcome. Now, one might imagine that stimulation means an increase in excitatory activity for the neurons at the tip of the electrode, but that isn't necessarily the case. What we see with deep brain stimulation is a change in the firing patterns of circuits. So in this illustration, we see patterns of activity recorded in the basal ganglia prior to turning on the deep brain stimulator. During stimulation, there is obviously an increase in activity, but really fundamentally a change in the pattern. And this change in the pattern of activity in the circuitry of the basal ganglia seems to be what allows for the normalization of motor function. So there's a lot of ongoing study of deep brain stimulation, really trying to understand what are we doing. Are we driving excitatory output? Are we driving inhibitory input? Are we just adding noise to the system? Or are we depolarizing the tissue at the tips of the microelectrodes to the point where the sodium channels are inactive and signaling is lost? Well, I think we're probably doing all of the above. And the point is that it need not matter that we target one particular type of physiological effect. What really matters for the patient is whether there's freedom of movement. And the goal of deep brain stimulation is to normalize patterns of activity in the basal ganglia to facilitate the expression of movement. And thankfully, many people around the world have been helped with this technology. And I would encourage you, if you have access to YouTube or other social media outlets, to go and look at some of the stories of these people who have volunteered their stories and videos of their lives to share with the world. And many of them are just really wonderful personal stories of triumph. Unfortunately, this approach doesn't work for everyone, and so we need to be cautious. We need to recognize that this is an invasive procedure. It has risk involved, and not everyone can be helped. So, as some of my colleagues in neurology here at Duke like to say, that deep brain stimulation is not going to cure Parkinsonism, but what it will do is it will increase your level of function if you are afflicted with the disease. And together with pharmacological approaches, behavioral approaches, exercise, deep brain stimulation might actually be an important adjunct to a more multidisciplinary approach to intervention. Now let's travel down the continuum and consider the role of the basal ganglia in creating hyperkinetic movement disorders. And the disorder that I'd like to use to illustrate this end of the continuum is Huntington disease. So Huntington disease is characterized by involuntary movements of the body, often involving the limbs, and because the flailing movements of the limbs resembles a dance-like posture or dance-like movements, this is called chorea, which is a Greek word that means dance. So involuntary choreic movements are characteristic of Huntington disease. Along with the disease that tends to set in the 4th and 5th decades of life is a severe dementia that can develop. And this dementia obviously can be quite tragic, and together with the unwanted movement that's being expressed can lead to great suffering on the part of the individual afflicted with the disorder, as well as that person's loved ones and system of support. As I mentioned, the disorder tends to first manifest itself by the 4th or 5th decade of life, and unfortunately most patients succumb to this disorder within about 10 to 15 years of the first appearance of symptom. Now, the pathology associated with Huntington disease actually is now traced back to a genetic origin. Huntington disease reflects an autosomal dominant mutation that results in the production of a gene product from a gene called Huntingtin, and this product contains a repeating sequence of CAG nucleotides. So most of us have a string of repeats, maybe around 20 or so repeats of this CAG motif in our Huntingtin gene. But if the number of repeats exceeds about 30, 35, 40, then the likelihood of developing Huntington disease increases dramatically. Now, we don't quite understand yet the link between this increase in these repeated motifs in the gene and the product of the gene and how that relates to the pathology in the brain. But what we do know is that with Huntington disease, there is widespread neuronal degeneration, and among the first structures to experience degeneration are the medium spiny neurons of the striatum that express D2 dopamine receptors. And that would be the neurons that give rise to the indirect pathway in the basal ganglia. So the consequence of Huntington disease is a loss of this indirect pathway from the striatum to the pallidum. So this means that we lose the ability to suppress movement in the following way. With the loss of this inhibitory projection, now there's way too much activity coming out of the external segment of the globus pallidus. That is going to lead to a dramatic reduction in the amount of inhibition that is being expressed by the internal segment of the globus pallidus in the motor thalamus. With insufficient inhibition of the motor thalamus, now we have increased excitation of this connection from the motor thalamus to the motor cortex. That's where the unwanted movements are coming from the insufficient suppression of the motor thalamus. Welcome to this tutorial on the means by which the cerebellum serves to modulate movement. This topic pertains to several of our core concepts. You're tired of hearing me say this, I know, but once again, I need to remind you that the brain is indeed the body's most complex organ, and that much of the basic function that we are learning about, especially in this unit of the course, pertains to circuitry that is under genetic determination. Now, that's not to say that this circuitry cannot be modified through our experiences in life. Indeed, that's the case. And as we come to the cerebellum, we come to a part of the brain that is especially involved in responding to our life experiences. And in a different tutorial, I hope to show you a rather dramatic example of one such paradigm that induces a dramatic change in the function of the cerebellum that we can watch happen in real time. So stay tuned for that. So I'm going to break this tutorial down for you into 3 parts. In this first part, I'd like to speak to you about just the basic parts of the cerebellum and provide for you an overview of cerebellar function. And then in the next part, we'll look more deeply at the circuitry of the cerebellum, including the inputs and outputs of the cerebellum. And then finally, we'll talk again at the end about cerebellum function. And then I'll talk to you a little bit about the signs and symptoms associated with dysfunction of the cerebellum. So our learning objectives for this part of the tutorial are, first of all, to identify and to discuss the basic parts of the cerebellum.
And I want you to be able to characterize in very general terms the major functions performed by the cerebellum. Now, before we say too much about the cerebellum in terms of its structure and function, I want to just highlight what has been sometimes called over the years the enigma of the cerebellum. And the enigma is that the cerebellum is one of the brain's most recognizable structures, and yet its function has been mostly elusive over these many decades that modern neuroscience has now been trying to discover the functions and operations of cerebellar circuits. It has a beautiful circuitry, as I hope to show you shortly, but the functions have been much more difficult to understand. Part of the enigma is that we know, unfortunately, from people that have had damage to the posterior part of the cranium, that destruction of the cerebellum leaves one still able to walk, to talk, to think, to see, to touch, to feel, to have a more or less normal emotional or cognitive life, but yet there is dysfunction. And so the enigma has been, how can such a massive part of the brain be damaged and produce seemingly only modest impacts on neurological function? Well, I think we perhaps know a little better now. Certainly we know more about the function of the cerebellum, but I think we're a little bit more sophisticated in our analysis of human performance and certainly in clinical care. So I think what we would say now is that yes, the impact of cerebellar dysfunction can be subtle, but it can be quite significant, and it certainly can have a significant impact on one's sense of self and one's sense of engagement in the communities in which we live. So there is a role for the cerebellum that's important, and it's one that we need to understand. And that will be the focus of the third part of this tutorial. So just keep in mind, the cerebellum is a massive brain structure, yet its functions have been somewhat enigmatic to understand. Well, let me try to resolve this enigma as best I can by just discussing with you in broad overview the function of the cerebellum. Well, in a phrase, the function of the cerebellum is error correction. And when we think of what it takes to actually do something difficult or to learn a new motor task, acquire a new skill, perhaps perform a new piece on a musical instrument that you may or may not be very familiar with, or perhaps it's a new dance, whatever kind of skilled motor behavior you can think of, there is an element of motor learning that's involved. And essentially, the essence of motor learning is implementing error correction. So the way the cerebellum does this is that it integrates executive commands about the intentions of our movements with sensory feedback about the way our body is actually moving. And it's this integration of executive signal and sensory feedback that gives rise to moment-to-moment corrections of behavior and performance. These functions facilitate learning when the errors are numerous. And if you'll recall the last time you tried to do something challenging or difficult with the movements of your body, especially involving your distal extremities, you know that there are a lot of errors, but it's through the experience of those errors that we can accrue skill. And it's that motor learning process that engages the circuitry of the cerebellum. Now, one key point that I want you to appreciate about the organization of the circuits of the cerebellum, we'll come to this in the second part of this tutorial, is that the cerebellum does not engage the lower motor neurons directly. In this sense, it's similar to the basal ganglia, which also did not engage lower motor neurons directly. Rather, the output of the cerebellum is directed towards circuits of upper motor neurons. And one important means by which the cerebellum will do so is by interacting with the thalamic nuclei that send projection to the motor cortex. A little bit more on cerebellar function. This might help you. I know it helps me. I like to think of the cerebellum as an organ of agility. And I want you to think about agility in the broadest possible terms. Think about anything that you can do with your body that you consider to be agile. Or maybe it's not movement of the body. Maybe it's movement of the mind. Maybe it's movement of thought. Maybe it's problem solving. Maybe you're really good at solving crossword puzzles, or you're very gifted in mathematics, or you're just a very logical, inductive sort of person, and you can reason your way through challenging mental problems. If that's you, then probably the cerebellum has contributed to your capacity to move your body or to move your mind with agility. So we want to account for how is this possible? Well, with respect to movement of the body, one component of agility involves the coordination of multi-jointed movement. So movement across multiple joints, often in a precise sequence of movement, sometimes from proximal to distal, although there may be some movements that work in the other direction. Now, I'd like to extend this concept of agility into the domain of cognition and imagine that the connections that the cerebellum maintains with circuits that engage the prefrontal cortex also operate in the same way as those circuits that engage the motor cortex. And from this standpoint, perhaps it's useful to think about mental problem solving as a kind of coordination of sequence. Or if we want to think of the various components of problem solving as movement across multiple hinged joints, if you get the metaphor, then I think we can understand the cerebellum as being involved in the coordination of multi-jointed cognitive processes. That is, cognitive processes that articulate and that are sequenced in very much an analogous way as the movements across multiple joints of a limb. Okay, well, that's my introduction. I want you to understand some of this background as we now seek to identify the basic parts of the cerebellum. Well, there are really three basic parts of the cerebellum that we want to consider. There's the cerebellar cortex, which is all of this massive tissue that we see from the surface view of the brain. In addition to there being an outer cortex, very much like a cortex of the cerebrum, there are deep nuclei that are buried down in the depths of the white matter of the cerebellum. So if we were to peer down into the core of the cerebellum, right along the roof of the fourth ventricle, we would see some gray matter, and these are called the deep cerebellar nuclei. So the gray matter of the cerebellum is organized into two tiers of processing. There's an outer cortex, and then there's an inner mass of deep nuclei. While the cerebellum is connected to the brainstem via three large stalks of white matter, and we call these peduncles, the cerebellar peduncles. So the cerebellar peduncles are the third major component of the cerebellum that we want to orient ourselves to. And there are actually three peduncles that come together near the central lateral region of the cerebellum. Those three peduncles are this large, massive middle cerebellar peduncle, by far the largest of the three. And just on the superior aspect of that peduncle, we find a superior cerebellar peduncle, and that is this structure that is shown here in this purple color, the superior cerebellar peduncle. This is largely a pathway that gets information out of the cerebellum down just on the medial and inferior edge of the middle cerebellar peduncle. We find the inferior cerebellar peduncle. So the middle is the largest. It's conveying signals from the brainstem, specifically the pons, into the cerebellum. The superior is a way to get signals out of the cerebellum, and the inferior peduncle is a bit more bidirectional than the other two. It is dominated, I think, by inputs that are going into the cerebellum, mainly from the spinal cord, but there are also connections that are going into the cerebellum from the brainstem via this peduncle, and there are efferent signals coming out of the cerebellum through the inferior peduncle. So we'll have more to say about this when we talk about the inputs and outputs of the cerebellar circuits. Now I'd like to draw your attention to the illustration in panel A. So here in panel A, we have a view of the cerebellum as if it were possible to remove the cortex from the cerebellum and flatten it out. If we were to do so, we would find a convenient way to recognize three major functional subdivisions of the cerebellum that we want to discuss. Now, there are many different ways to divide the cerebellum. One could take an anatomical approach. I'm choosing to take for you a functional approach. And with this in mind, we would define a spinal cerebellum. The spinal cerebellum includes essentially a medial strip of cerebellar cortex that spans what we call the two cerebellar hemispheres and includes the continuity of those hemispheres along the midline region here, which has a memorable name. This is called the vermis, which is Latin for worm. It's as if we're looking down on a segmented worm. Unlike the hemispheres of the cerebrum, there really is no separation of the gray matter here between the cerebellar hemispheres. Rather, there's continuity. So this spinal cerebellum really is the central region along the midline and the parasagittal regions of the cerebellar hemispheres. And we call it the spinal cerebellum because this is the part of the cerebellum that is mostly concerned with the movements of the arms and the legs. It is receiving input from the spinal cord. Now, there's also some representation of cranial movement here in the spinal cerebellum, so let's not let that fact deter us from calling this the spinal cerebellum. The next component is this very large cerebrocerebellum, and this is what comprises most of the cerebellum that we observe when we look at the human cerebellum. We have these very large, very extensive lateral hemispheres of the cerebellum, much larger than any non-primate mammal and perhaps even as large or larger than any other of the primates. So this is the part of the cerebellum that is involved with circuitry from the cerebral cortex that returns a signal back to circuitry of the cerebral cortex. Hence its name cerebrocerebellum. We'll say more about the functions of this division of the cerebellum in the third part. For now, suffice it to say that the cerebrocerebellum is concerned with the skilled movements that we perform with our distal extremities, especially when then there's some kind of skill that is being executed according to some goal-directed motor plan. And then the third component of the cerebellum that I would mention pertains to this little structure that is tucked away under the ventral and anterior aspect of the posterior cerebellum. This is called the vestibulocerebellum. And this term vestibulocerebellum is a functional term. Anatomically, it relates to these two parts of the cerebellum that we call the flocculus and the nodulus, and together we call this the floccular nodular lobe. So the vestibulocerebellum is the floccular nodular lobe, and this is the part of the cerebellum that is intimately connected with the vestibular nuclei of the dorsolateral tegmentum in the upper medulla and the caudal pons. So this part of the cerebellum is going to have much to do with calibrating and governing the motor output from our vestibular nuclei. Let me just show you a bit more closely these functional divisions with a special attention now given to the organization of the representation of the body in the spinal cerebellum. As I mentioned just a moment ago, the spinal cerebellum refers to this central strip of cerebellar cortex that includes the vermis right along the mid-sagittal plane and a paramedian strip extending out maybe a centimeter or so from that vermis. And here we have a map of the body. And this body map is not a single smooth continuous map, but it's fractionated in a way that's not unlike the mapping of motor intention in the motor cortex is fractionated. In fact, we have multiple representations that are present here, and this is a very simplified view and undoubtedly an incorrect view with respect to many of its details, but it conveys a sense of the organization of the representation of movement in the spinal cerebellum. And what I would especially highlight is the fact that the axial representation is closer to the middle of the spinal cerebellum along the region of the vermis, whereas the representation of the distal extremities extends outward from the vermis into this paramedian strip of cerebellar cortex that begins to extend into the lateral hemispheres. Now, as I mentioned, there are two levels of processing with respect to the gray matter of the cerebellum. There is the cortex, and then there's the deep nuclei. So the cortex is what we see from the outside when we look at these massive cerebellar hemispheres. And this cortex is folded into these tight little mini convolutions. Now in the cerebrum we call these gyri, and the spaces between them we call these sulci. In the cerebellum we call these folia, because if one were to look in cross-section at this cortex, what one would see is a nice delicate extension of processes, almost like leaves on a tree. So folia implies leaf, and what we see in the cortex of the cerebellum looks more like leaves than it does a large massive gyrus. Now what I would also point out about these folia is that they extend over quite a considerable distance. One can follow the same fold across the cerebellar hemisphere for at least several centimeters, perhaps even through the spinal cerebellum, across the vermis, and into the opposite cerebellar hemisphere. So when we talk about the circuitry in detail, we'll see what accounts for this organization of these folia that extends across the long axis of the brainstem in this way. Now below the cortex there are the deep nuclei, and the deep nuclei are the structures that are highlighted here. They actually reside in the white matter just on top of the roof of the fourth ventricle. There's a very large structure that resembles sort of a row of molar teeth, and because of its resemblance to dentition, this nucleus is called the dentate nucleus. That's by far the largest of our deep cerebellar nuclei, and it's a nucleus that seems to be mainly related to the cerebrocerebellum. Right next to the dentate nucleus are at least a pair of nuclei, sometimes more depending on how you subdivide. These are called the interposed nuclei, and these nuclei pertain mainly to the lateral aspects of the spinal cerebellum. And then most medially there is a small nucleus called the vestigial nucleus, and this nucleus pertains mainly to the vermis, or the medial region of the spinal cerebellum, and it's found appropriately on the medial side of this cluster of nuclei. Now you may be wondering about a deep nucleus for the vestibulocerebellum. As I'll mention, the deep nuclei of the vestibulocerebellum are actually the vestibular nuclei of the brainstem. So from this standpoint, we can consider the vestibular nuclear complex as deep cerebellar nuclei that are displaced out of the cerebellum and into the brainstem. Well, you'll understand the anatomical relationships between cortex and deep nuclei as we get into part 2 of this tutorial. At this point, I actually want to show you a bit of histology. So this is a transverse section through the brainstem, and our inset below hopefully orients you to what you're looking at. We're seeing a cross-section through the caudal part of the pons that extends through the fourth ventricle and into the cerebellum. And what I want to highlight for you is this beautiful dentate nucleus. And hopefully you can imagine why the early anatomists called it a dentate structure, given the resemblance, at least in your imagination perhaps, to a row of teeth. So that would be the dentate nucleus. It looks like we have elements of our interposed nuclei here, and then this is our vestigial nucleus. So these are the nuclei that are arranged in the base of the cerebellum in the white matter on the dorsal and lateral aspects of the fourth ventricle. Now I'll just caution you, if you look carefully at this dentate nucleus, you may think that you've seen something like that before if you've perused through sections of the brainstem in the medulla. This dentate nucleus visually resembles the inferior olivary nucleus. We'll talk about the inferior olivary nucleus in just a little while. So I don't want you to confuse the two. Even though they look similar, we know that this is the dentate nucleus because it is in the cerebellum, right? It's in the white matter at the base of the cerebellum. It's dorsal and lateral to the fourth ventricle. whereas the inferior olivary nucleus is in the anterior aspect of the brainstem in the medulla, which is a few centimeters in the inferior direction to where this section has been taken. Well, let me show you one other view of this system. This is from a rather unique section prepared by one of our histologists from a generation ago here at Duke. This is a section taken parallel to the long axis of the brainstem, and it happens to catch the base of the cerebellum as it's connecting to the brainstem. So this is a myelin stain, as was the previous image. So the white matter is going to be dark and the gray matter is going to be light. And what we see is the cerebellum out in this region of the image. So we have a cerebellar cortex and white matter underneath, and then now here's our beautiful dentate nucleus. Very rich, robust dentate nucleus as seen in this section. And we'll come back to this, but I'll just highlight for you this white matter that's streaming out of the dentate nucleus. These are axons that are going to be forming the superior cerebellar peduncle. And what these axons do is they stream out of the dentate nucleus, they run through that peduncle into the pons, they cross the midline, and then they synapse in targets in the thalamus and in the brainstem. So more on that when we talk about the outputs of the deep cerebellar nuclei. Okay, we've concluded our introduction to the basic parts of the cerebellum. When we come back, we'll take a closer look at cerebellar circuits. Welcome back to our tutorial on the cerebellum, and in this part I'd like to take a closer look at the circuitry of the cerebellum. And my learning objectives for you are that I want you to be able to make a sketch of the major inputs and outputs of the cerebellum. So, as all sketches that I encourage you to make, the bigger the better. As you're making your sketch, I want you to be able to have a discussion, perhaps with a friend, a family member, maybe someone who's taking this course along with you. But have a discussion, even if it's just to hear the sound of your own voice. I would encourage you to describe the circuitry that's involved in the processing that runs through the cerebellum. And as I'd like to describe for you, we can conceptualize this circuitry as involving a main excitatory loop that runs through the deep nuclei of the cerebellum, and then an inhibitory side loop that runs up through the cortex and then back down to the deep cerebellar nuclei before output leaves the cerebellum to influence circuits of upper motor neurons. Well, before we talk in detail about those circuits, I want to give you just a broad overview of the inputs to the cerebellum and then talk just a bit about function. So the main inputs from the cerebellum come from a couple of different sources. They come from the pontine nuclei. These would account for the majority of the inputs to the cerebellum. And notice the relationship of the pons to the cerebellum. The input from the pontine nuclei crosses the midline of the brainstem and projects into the contralateral hemisphere of the cerebellum. And the pathway by which it crosses the midline and enters the cerebellum is the middle cerebellar peduncle. Now notice that these pontine nuclei are in turn supplied by inputs that are derived from the cerebral cortex. So the cerebral cortex then has the means by which it can send signals into the cerebellum, but only indirectly through a relay in the pons. And as we discussed in the opening to the first part of this tutorial, one important function of the cerebellum is to integrate executive commands about how we intend to move our body. Well, this is how this happens. Through input from the cortex to the pontine nuclei that are at the base of the pons, there's a synaptic connection and then a relay from the base of the pons into the contralateral cerebellum. And notice that the input is being distributed both to the cortex of the cerebellum as well as to its deep nuclei. Well, that's one massive and very important source of input to the cerebellum. Another important source of input to the cerebellum comes from our sensory systems, and specifically from our proprioceptive systems concerning the movements of our arms and legs, and this is fed into the cerebellum via relays from the spinal cord and the caudal part of the medulla. And then there are inputs concerning the movements of our head. And these are conveyed into the cerebellum via the vestibular nuclei of the dorsolateral tegmentum of the upper medulla and the caudal pons. And at least for some ganglion cells in the vestibular division of the eighth nerve, there may be direct connections from the eighth cranial nerve into the cerebellum. So the cerebellum is getting this very important sensory feedback about how we're moving our bodies with respect to spinal cord inputs, and then how our head is moving through three-dimensional space. Well, there's a third major source of input to the cerebellum that I want to mention, and it's coming from a really fascinating nucleus of the ventral and somewhat intermediate aspect of the medulla. It's called the inferior olive, or the inferior olivary nucleus. This nucleus sends axons across the midline that enter the cerebellum from below via the inferior cerebellar peduncle. And the inferior olive is providing a learning signal for the cerebellum. And we will talk about this in some detail, but essentially what this signal is doing is it's conveying to the cerebellar cortex a time for synaptic change. And this synaptic plasticity appears to be the cellular and synaptic basis for motor learning in the cerebellum. Now, this inferior olive, in turn, is getting inputs that are descending from a pathway derived from the cerebral cortex. And the relay in this pathway is the red nucleus. So notice that the cortex is sending its executive signals into the cerebellum via a relay in the pons, but it's also informing the red nucleus, which is in turn activating the inferior olive. And when the inferior olive is active, now the cerebellum will engage in synaptic plasticity, and we think produce an error correction signal that can improve performance. Now let me say just a few more words about the organization of these inputs before we look at a more fine-grained level at the organization of the circuits in the cerebellum. So we have some anatomical terms that I want to define for you. They reflect the names the anatomists have given to the appearance of the inputs that we see under the microscope when we look at the cerebellum, at the cortex, and at the deep nuclei. What we see when we look at the inputs that are coming from the pons and those that are coming from the spinal cord and the vestibular nuclei, what we see is a morphological appearance of the input that we call the mossy fiber. So these inputs give rise to what we call mossy fibers. And this is now a bit of a shorthand term to describe all of this afferent input that is converging on the circuitry of the cerebellum, all except for one very special kind of input. And that is the input that's coming from the inferior olive. This one type of input we call the climbing fiber. And the reason we call it the climbing fiber should be obvious in just a few minutes. It has to do with the intimate relationship of the axons of this inferior olivary neuron with respect to the principal cell type that we find in the cortex of the cerebellum. Now, let me emphasize that both the mossy fibers and the climbing fibers terminate at both levels of cerebellar processing, in the cerebellar cortex and also in the deep nuclei. Now, perhaps it will be helpful just to remind you a bit of the inputs to the cerebellum with a little bit more detail. You will recall that the inputs to the cerebellum from the spinal cord convey signals about the movements of our body that are sensed by our proprioceptors, and they are conveyed via pathways that serve the lower extremities and the upper extremities. The pathway that serves the lower extremity involves a relay in the dorsal nucleus of Clark, which is found in the thoracic segments of the spinal cord. The dorsal nucleus of Clark grows an axon that runs up the dorsal lateral white matter of the spinal cord and forms a tract that we call the dorsal spinal cerebellar tract. This is the major relay of proprioceptive signals from the lower extremities into the cerebellum. This pathway runs on the ipsilateral side of the cerebellum and the spinal cord. So the cerebellum is getting information about the movements of the ipsilateral side of the body. The same is true for the upper extremity, and this pathway runs from the first order neuron in the spinal nerves that enter the dorsal column, and then there's a synapse in the external cuneate nucleus, the most lateral of our dorsal column nuclei, and from there the second order neuron projects into the cerebellum via the inferior cerebellar peduncle in parallel with that dorsal spinal cerebellar tract. And the cuneocerebellar tract is now involved with conveying proprioceptive signals about the movements of the upper extremities into the cerebellum. So that's what we find here in the input from the spinal cord. So this input from the spinal cord is going to be directed again mainly at that median zone of the cerebellum that we call the spinal cerebellum. The cerebrocerebellum, on the other hand, is going to get information that is principally relayed via these pontine nuclei. So this massive connection from cortex to pontine nuclei to cerebellum is mainly going into the lateral hemispheres, into that region that we call the cerebrocerebellum. And then these vestibular inputs that are being sent in some cases directly from the eighth nerve into the cerebellum, and others in a relay through the vestibular nuclei of the brainstem, these are being conveyed to the vestibulocerebellum, that floccular nodular lobe that's tucked underneath the posterior part of the cerebellum. One final point to make about the organization of this circuitry that should have been evident to you as we've been talking about it, and that is that this cerebellum is representing the ipsilateral side of the body. So perhaps I'm belaboring this point just a bit, but it certainly is worth some added emphasis. Perhaps the spinal cord is what first sets up this ipsilateral representation by virtue of its ipsilateral dorsal spinal cerebellar pathway that runs in via the inferior cerebellar peduncle. But this principle of ipsilateral representation in the cerebellum creates a bit of a problem, does it not? Because you now understand that when it comes to the cerebral hemispheres, the principle is contralateral representation. So this means that one side of the body is going to be represented in the opposite cerebral hemisphere, but in the same-sided cerebellar hemisphere. And perhaps this provides a helpful way to just think through the necessity of this connection from cortex to pons and then from pons to cerebellum. This will also be a challenge when thinking about the outputs of the cerebellum that provide a means by which the cerebellar hemisphere on one side of the midline can relate to circuits of upper motor neurons on the opposite side. So we'll face that challenge when we see an analogous figure that will be illustrating the output pathways of the cerebellum. But with respect to input, we can see how the spinal cord seems to set up this ipsilateral representation, and then the relay from the cortex meets the appropriate constraints, allowing one side of the cerebrum to interact with the appropriate hemisphere of the cerebellum. Okay, now let's have a deeper look into the cortex of the cerebellum, and I want to tell you about some of the important neurons that we find there. The principal neuron that we find in the cerebellum is a very large neuron called the Purkinje cell, and it's really one of the most beautiful neurons that you'll ever want to see under the microscope. It's a very large cell body that gives rise to these incredibly complex branching patterns of dendrites, and this illustration of course cannot do justice to the form and the beauty of the Purkinje cell and its dendritic arbor. Now this Purkinje cell is the principal integrator of inputs that are coming into the cerebellar cortex, and it's receiving a massive input from the axons of these really tiny cells that account for the majority of the cells that we find in the cerebellum. The cells in question here are the granule cells of the cerebellum, very small types of neuronal cell bodies that grow an axon out towards the outer part of the cortex of the cerebellum, and then that axon splits into two parts. And now these two parts can grow for some considerable length in parallel along a folium that forms the folding of the cerebellar cortex. So these are called parallel fibers because they are growing in parallel to one another and parallel to the organization of the folia of the cerebellum. Now, it's difficult to appreciate this fact in the illustration, but the organization of the Purkinje dendrites are orthogonal to the parallel fibers that are running through them. In fact, if one were to look at a Purkinje cell, one would find a remarkable geometry to its dendrites. While most neurons in the brain grow dendrites in all different directions, the Purkinje cells grow their dendrites in a very narrow plane, much like the fingers of my hand are extending in a narrow plane. Well, I brought a little bit of a visual aid that hopefully will illustrate for you what these Purkinje cell dendrites actually look like. What I've brought to show you is my Purkinje neuron of the sea, as I like to think of this. Well, actually, no, this is not a Purkinje neuron. This is a sea fan, which is a type of coral that grows in the ocean waters. And what's distinctive about this sea fan is that the arborizations of its dendritic structure is restricted to a very narrow plane, in the same way that the dendrites of the Purkinje cells are constrained in a very narrow plane. Now, what you have to imagine is this single plane is orthogonal to this massive system of parallel fibers. In fact, this input of parallel fibers to the Purkinje cells provides the most extreme form of convergence that we know about in neural circuits. Perhaps as many as 100,000 or even 200,000 individual parallel fibers will be running through this beautifully intricate lattice work of dendrite that is arranged to sample these massive parallel fibers that are running through it. Let me tell you just a little bit now about the inputs, and then that will get us into a bit of the neurochemistry and the physiology of how this circuitry works. Now, the granule cells are getting input from mossy fibers. So here's a mossy fiber that's growing in, and it's making synaptic contact in a very particular morphological structure that we call a glomerulus here. And there are other interneurons involved as well that I won't take the time to mention. For now, I'll just emphasize that the mossy fiber, and again, that's all the inputs to the cerebellum except for that special inferior olivary input. These mossy fibers are synapsing on the dendrites of the granule cells, and this is an excitatory synapse, so the mossy fiber is exciting the granule cell. The granule cell gives rise to these massive parallel fibers that make an excitatory synaptic connection onto the dendrites of the Purkinje neuron. So the granule cell parallel fibers then are exciting the Purkinje cells. Well, the Purkinje cells send their axons down through the white matter where they synapse on cells in the deep cerebellar nuclei. Now, the synapse between the Purkinje cell and the deep cerebellar nucleus is an inhibitory synapse because the Purkinje cells release GABA as their neurotransmitter, and they interact with receptors that mediate an inhibitory influence of that neurotransmitter in the deep cerebellar nucleus neuron. So notice we have two excitatory connections in series and then an inhibitory connection, which can fundamentally alter the way the deep cerebellar nucleus integrates its own input. One final special input to highlight here in this illustration, and that would be the climbing fiber. So the climbing fiber is again derived from the inferior olivary nucleus, and now in this figure we get a sense of why it's called the climbing fiber. This axon makes contact with a single Purkinje cell and then wraps its axons around the proximal dendrites of these Purkinje neurons with intimate contact, suggesting a very powerful synaptic relationship between this climbing fiber and the Purkinje cell. Indeed, it is one of the most powerful synapses that we know in the mammalian brain. Okay, let me take you through this circuitry again in a somewhat schematic way and try to help you understand the nature of the computations that might be going on here. So let's look at the illustration that shows us the neurons and the synaptic inputs and talk once again through the connections that link the inputs to the cerebellum to the cortical circuits. So the inputs to the cerebellum in the form of the mossy fibers come and they make synaptic connections with the granule cells. They also send branches down into the deep cerebellar nuclei and they release excitatory transmitters. Now the granule cells give rise to these long bifurcating axons that form these parallel fibers, which then make synaptic connections with the Purkinje cells. And then the Purkinje neurons integrate those parallel fiber inputs and then send their axons down to the deep cerebellar nuclei where they release gamma-aminobutyric acid, GABA, onto the deep cerebellar nucleus neuron. Meanwhile, there is a climbing fiber involved. So the climbing fiber gives rise to inputs at both the level of the deep nuclei and also, of course, this very special intimate relationship between the climbing fiber and the proximal dendrites of the Purkinje cell, where there's this massively strong excitatory connection. So we'll come back to this when we talk about plasticity in just a moment. But to put all this in action, I would suggest that we conceptualize these connections as consisting of a deep or a main excitatory loop and a cortical inhibitory loop that runs through the Purkinje neuron and back down to the deep cerebellar nucleus. So here's what I have in mind. There is excitatory input that comes into the deep cerebellar nucleus, and meanwhile there is a transformation of that excitatory input into an inhibitory signal that can then modulate the output of that excitatory neuron from the deep cerebellar nucleus. And as we'll emphasize, the output from the cerebellum really comes from this deep nucleus. Okay, so the Purkinje cell then is in a position to chop or to modulate the activity of that deep cerebellar nucleus neuron. And the way motor learning seems to work, and the way synaptic plasticity in the cerebellum seems to work, is to actually modify the strength of this inhibition that is conveyed from the Purkinje cell down into the deep cerebellar nucleus. Now, knowing that this is an inhibitory connection, if one imagined that motor learning is consistent with a stronger output from the cerebellum, one way to provide that stronger output would be to weaken this input coming down from the cerebellar cortex. So if we were to reduce that synaptic inhibition, we'd have the effect of actually strengthening this connection by which the deep cerebellar nuclei influence the operation of upper motor neuron circuits. Well, let's now have a look and see how this actually happens. So here is a view of the circuitry, again, with a bit more realism attached to the dendrites of the Purkinje cell and the relationship of the parallel fibers to those dendrites as well as the climbing fiber. Now imagine it were possible to do the following experiment, where we can record from the dendrites of a Purkinje cell and measure the postsynaptic response following activation of parallel fibers and climbing fibers. And if we do an experiment such that we combine low frequency stimulation of the climbing fiber at the same time that we activate the parallel fibers, what we find is that there is a depression of the strength of the synapse between the parallel fiber and the Purkinje cell that is expressed over the next 10 to 20 minutes or so and can persist for a very long time. So the form of synaptic plasticity that we see here in the cerebellar cortex is long-term depression. And that can be measured here in this experimental record where there's a recording of the excitatory postsynaptic amplitude at the parallel fiber Purkinje cell synapse. And with conjoint stimulation of both climbing fiber and parallel fiber, now we see over time a decline to about 50% or so of the amplitude of that excitatory postsynaptic connection. The means by which this synapse is weakened should be somewhat familiar to you, although there are a few surprises in the metabolic cascade that leads to activation of these synaptic connections and the weakening of this synapse. The part that should be familiar to you is the notion that one way to weaken a synapse would be to internalize AMPA receptors. And indeed, this is the molecular mechanism that expresses long-term depression at this synaptic connection. The trigger that induces this internalization is perhaps a bit unexpected. It involves glutamate, as have our other mechanisms of synaptic plasticity that we've discussed, except the NMDA receptor does not appear to be involved in this story. Rather, the receptor that mediates this phenomenon is a metabotropic glutamate receptor. And glutamate is released at the parallel fiber synapse. It interacts with this metabotropic receptor for glutamate, and this engages a cascade of second messenger systems that liberate calcium from intracellular stores. So there is a sudden increase in calcium following the activation of these second messenger systems. But that release of calcium doesn't seem to be sufficient to cause robust internalization of AMPA receptors. So we need to get more calcium into this postsynaptic spine rapidly. And the way that happens is through activation of our climbing fiber system. So with the activation of the climbing fiber, there will be a massive depolarization of this dendrite. Again, the climbing fiber synapse is one of the strongest synapses that we know about within the central nervous system. So when the climbing fiber is active, there's this profound depolarization which will open up voltage-gated calcium channels, allowing an extra bolus of calcium to flood into that postsynaptic spine, further elevating the levels of calcium that we find. And once a sufficient amount of calcium enters that spine, then yet additional second messenger systems will be engaged that lead to the activation of phosphokinase C and other substrates of phosphorylation that lead to the internalization of these AMPA receptors and the reduction in synaptic strength. Now, please don't be confused about the mechanisms of plasticity that we've discussed so far. Just recognize that the molecular mechanisms that mediate plasticity in the cerebellum are distinct from those that we talked about in the cerebral cortex. In the cerebral cortex, high calcium leads to long-term potentiation, but in the cerebellum, high calcium leads to long-term depression. All right, I'd like to switch gears a bit now and talk to you about the anatomy of the output of the cerebellum. And we'll consider this anatomy really in two steps. First, I'd like to talk to you about the ascending outputs from the cerebellum. They're ascending anatomically in that they leave the cerebellum and they enter the brainstem via the superior cerebellar peduncle, and then they ascend in the superior direction to encounter targets in the brainstem and in the motor thalamus. So the source of these outputs are the deep cerebellar nuclei, specifically the dentate nucleus, and perhaps the interposed nuclei as well. But the dentate nucleus quantitatively is the largest and most significant of the sources of these ascending cerebellar outputs. So recall again that we've got a bit of a challenge, that we've got one side of the cerebrum is related to the opposite cerebellum. So for the outputs of the cerebellum to impact these upper motor circuits, there needs to be a crossing of the midline, a decussation. And here's how it happens. The deep cerebellar nuclei in question, namely the dentate nucleus, grows an axon that exits the cerebellum through the superior cerebellar peduncle. And some of these axons will ascend and terminate in the ventrolateral complex of the thalamus, the ventrolateral ventrally anterior nuclear group. There are other axons that will terminate among upper motor neurons in the superior colliculus. Probably not the same axons, but nearby axons. So these ascending inputs target the motor thalamus and the superior colliculus. There are also collaterals that will reach yet a different target, the red nucleus. Pardon me for writing the word red in blue, but the red nucleus also receives collaterals from these axons as they make their way through the midbrain on their way to the thalamus. Well, the motor thalamus then, as you know, will send its inputs to the primary and the premotor cortex. And at this level, we believe that the error correction signals can modulate the output of the motor cortex. Here's an anatomical view of these outputs, and draw your attention again to our beautiful dentate nucleus here. So there are cells that sit in this dentate nucleus that grow axons that stream out across this beautiful superior cerebellar peduncle, which is this big bundle of white matter that we see here and there. So we'll call that superior cerebellar peduncle. And notice that the decussation is actually outside of the cerebellum in the upper part of the pons, near the junction of the pons in the midbrain. And the axons branch and terminate. Some of them will in this red nucleus, which we find here in the midbrain, and then others will terminate up around in this region, the area of the ventral anterior ventrolateral complex of the thalamus. So this decussation then allows the appropriate hemisphere of the cerebellum to relate back to the upper motor circuits that govern the same part of the body. Now let's talk about what we might call the descending outputs of the cerebellum. Anatomically descending, not up to the motor thalamus, but directed towards brainstem targets. So recall again that we have our deep cerebellar nuclei. Near the midline, we've got the vestigial nucleus, and a bit more laterally, we have the dentate and the interposed nucleus. And for the descending inputs now, we focus mainly on the connections to the reticular formation of the brainstem. Now, the vestigial nucleus also has some connection to the superior colliculus, but it's probably not as functionally significant in our brains as is the larger output that's coming from the dentate nucleus. So we'll focus mainly on the right side of this figure to consider these descending outputs. So again, the major output of concern here is connections from the medial part of the deep nuclei of the cerebellum to the reticular formation of the brainstem. And the reticular formation, as you know, give rise to projections down to lower motor neurons and the medial part of the ventral horn of the spinal cord. So these are motor neurons that are going to be mainly concerned with posture and the movements of the proximal muscles of the limbs. Now, I mentioned in our introduction that the vestibular nuclei are sometimes considered deep cerebellar nuclei that are displaced out of the cerebellum and into the brainstem, and here's why. The vestibular nuclei receive input directly from Purkinje cells of the vestibulocerebellum. And these nuclei then are in a position that's somewhat similar to these deep nuclei. Although in this figure we chose to put them outside of the cerebellum as they actually fall anatomically, right alongside the reticular formation neurons. So these vestibular nuclei then give rise to their own spinal projections, where they helped to maintain balance under conditions in which there's been some kind of acceleration of the head, often associated with a disturbance of postural control. Well, when you come back for part 3, we'll talk about functions and dysfunctions of the cerebellum. Welcome back to part 3 of this tutorial on the cerebellum. I'd like to say a little bit more about the functions of the cerebellum and then talk to you some about dysfunctions of cerebellar circuits. So my learning objectives are that you would discuss the means by which circuitry in the cerebellum aids to increase the success of volitional motor performance. And I want you to be able to describe the clinical signs and symptoms associated with cerebellar damage. Well, let me first summarize both the anatomy and also now the function of these cerebellar circuits. And I'll reinforce for you our functional division of the cerebellum into a medial spinal cerebellum, a lateral cerebrocerebellum, and an inferior lobe that we call the vestibulocerebellum. So beginning with the largest of these divisions, the cerebrocerebellum. The cerebrocerebellum includes much of the hemispheres of the cerebellar cortex that we can appreciate when we see the cerebellum in the human brain. And these hemispheres send connections into the dentate nucleus down in the deep white matter of the cerebellum. The dentate nucleus sends its output through that superior cerebellar peduncle up to the circuitry of the motor thalamus that engages our premotor and motor cortex. And we think this part of the cerebellum is especially concerned with coordinating, organizing, and perhaps even planning the skilled execution of behavior that we do with our hands and our feet. So the connections suggest that this may be the case because the output of the dentate nucleus is largely engaging circuits that will impact the functions of the premotor cortex. The spinal cerebellum sends its inputs down to the more medial parts of the deep cerebellar nuclei, engaging the interpose and the vestigial nuclei. The interpose nuclei are growing connections that impact the motor thalamus and can, in turn, directly affect the output of the motor cortex. But the interpose nuclei, together with the vestigial nuclei, are also growing axons into the brainstem reticular formation. So we think that together these inputs are governing the execution of behavior more so than the planning for the skilled performance of what we do with the distal extremities. So it's a fine distinction there, perhaps one not worth making, but at least anatomically speaking, we associate the spinal cerebellum more with the control of, let's say, the more proximal parts of our limbs together with our postural muscles and our muscles that we use for locomotion, whereas the cerebrocerebellum we associate with the activities that are guided by the premotor cortex that are especially involved with the skilled behaviors that we do with our hands and our feet. And that leaves us with the vestibulocerebellum, that floccular nodular lobe that is directly connected with the vestibular nuclear complex of the brainstem. This nuclear complex, as you know, is mainly concerned with the feedback, adjustment of posture, given signals that are associated with the accelerations of the head. And these adjustments typically help to stabilize our posture against some unanticipated disturbance. So this has been a summary of the anatomy that gives you a sense of what aspects of motor control the cerebellum might have an influence over. Let me give you a little bit more of a sense of exactly what that nature of that influence might be. So when you think about the function of the cerebellum, I want you to think of coordination. The coordination of sequenced movement or multi-jointed movement when it comes to our extremities. When it comes to those domains of planning, perhaps even those domains of reason and problem solving, we can think of the influence of the cerebellum as helping to coordinate the sequences of events that are necessary for planning an act or for solving a problem. And the key operative word is coordination. So the cerebellum, as it coordinates these activities, it can help to improve the stability of our actions as well as their fluidity. The cerebellum has a role to play in dampening down the oscillations that might have as flexor and extensor muscles are coordinated by lower motor circuits. As the cerebellum is serving to correct errors in motor performance, it can also play a role in improving motor performance over time through the accrual of experience. And in this way, the cerebellum can assist motor circuitry in motor learning, because after all, correcting errors is exactly what is necessary for motor learning to occur. The cerebellum may not necessarily be a site where skill is stored, but it seems to be a necessary circuit that is engaged in the process of improving performance and thereby acquiring the skill in the first place. One of the most amazing facts of cerebellar-based plasticity that I've learned about is how specific that plasticity can be. We can do a pretty simple demonstration, and in fact I hope to show you this demonstration to make available for you a video recording of one of my experiments in action that involves plasticity and how our visual world is mapped onto our map of motor output or motor intention. And this would involve the task of throwing a ball at a target. And what we can demonstrate is that the plasticity that takes place, the learning that takes place under experimental conditions, can be specific for not only the motor structure that is being used, the arm of the thrower, but also the program itself, that is, the way that arm is used. So we can show plasticity of, let's say, an overhand throw, but not an underhand throw. Same muscles, same joints, roughly the same kind of sequence, but the details of how that multi-jointed movement is sequenced to perform an overhand versus an underhand throw differ. And it seems to be at that level of the organization of the motor program for movement that the motor learning takes place, or the adaptation occurs. So a remarkable degree of specificity in what the cerebellum is actually operating upon to change. Now, it's quite interesting to think that maybe something similar is happening in the domain of cognition. We know that the dentate nucleus sends connections not just to the motor thalamus, but also to more medial parts of the thalamus that send connections to the prefrontal cortex. This suggests that perhaps the coordination, that is the agility that the cerebellum provides for movement of body, might also apply to movement of mind. And one interesting clinical observation has been that patients with cerebellar injuries often fail in problem solving when it requires a sequential procedure. Perhaps you've seen some simple board games or peg games that require implementation of a strategy that involves moving in one step, followed by another, and another. Well, people with cerebellar injuries, while they are able to execute the motor steps that will be required of the game, what they seem to lack is the capacity to sequence through what one step will lead to and the scenario that the next step will provide. So it's a matter of sequence and coordination of our problem-solving faculty that seems to be impaired in patients with lesions in certain parts of the lateral hemispheres of the cerebellum. So I say that just to emphasize that the cerebellum coordinates movements of the body, and it also seems to assist in coordinating movements of the mind. So it should not be surprising that clinical populations with cerebellar injuries may have deficits in the cognitive domain as they may in the domain of motor control. So what are those deficits? Well, if the cerebellum is principally concerned with governing the ipsilateral side of the body, then we would expect deficits to be observed in movements of the ipsilateral side of the body. So the important principle for you to appreciate here is that the clinical signs of cerebellar lesions are always ipsilateral to that lesion. Now this stands in contrast to what you have learned about damage to upper motor neurons, where the clinical signs and symptoms are going to be contralateral to the location of the injury. As I've been saying, the cerebellum represents the ipsilateral side of the body, and perhaps this reflects the fact that the spinal cerebellar pathways are set up to provide ipsilateral input. Now, when it comes to cerebellar dysfunction, we would expect there to be an incoordination of movement. We call this ataxia. And ataxia can be further characterized by the clinicians. I won't get into that now, but just know that may be coming if you are getting deeper into the neurological sciences. But there are other aspects of this picture that I do want to talk about. One of them is intention tremor. So patients who have damage to the cerebellum, they tend to have tremulous movement when they are intending to move, such as if they were to reach out to grab an object, there is a tremulous movement that reflects an unsteadiness or an incoordination across those joints. There may be an instability of the limb as it approaches a target. It may undershoot a target, or it may overshoot that target if one were to reach out to grasp something. So there is a dysmetria, which refers to a lack of gauging the distance that is required to accurately execute a motor skill. There may be an impairment of alternating movements that is sometimes tested in the clinic. One should be able to alternately rotate around the wrist at a fairly rapid rate. And if there is either a breakdown in rhythm or a reduction in speed, then perhaps that suggests that there may be a problem in the governance of that motion via the cerebellum. And then finally, there can be a decomposition of movement. Rather than a smooth coordination across multiple joints in, let's say, a visually guided reach, there may be a breaking down of that motion into component parts. So there can be a decomposition of that smooth coordination or that smooth sequencing of action across these joints. I'd like to leave you with a short clip that we recorded in the lab when we encountered a brain that seemed to show evidence of degeneration of the anterior part of the vermis that is fairly characteristic in individuals that have abused alcohol for many years in their life. So one of the most common cerebellar dysfunctions that you may see if you're heading into the healthcare world is alcoholic cerebellar atrophy. And this can result in an impairment of the functions that are performed by the medial parts of the cerebellum, involving the vermis and the cortex just lateral to the vermis. This seems to be the region of the cerebellum that degenerates with chronic alcohol abuse for reasons that are not entirely understood. But when one thinks of this part of the cerebellum, I hope you should now recognize this as the spinal cerebellum, which is that portion of the cerebellum that is going to be especially concerned with the proximal limb muscles that are important for walking, for posture, and this is likely what explains the staggering gait that is characteristic of individuals that are chronic abusers of alcohol. They may be staggering not because they're inebriated, but because they've suffered this kind of medial and paramedial damage to their cerebellar hemispheres, as you are about to see.
Now, I want to show you the cerebellum from what is likely a normal brain in order to compare with this specimen that we have in front of us. So here we have a cerebellum from an individual who died without neurological complication. And I want you to notice how tight and full are these tiny little branches or folia that form the cortex of the cerebellum. In contrast, this other brain has an unusual amount of space between the folia of the cerebellum. These spaces are indicative of one particular pathology that seems to result in the atrophy of this anterior and medial part of the cerebellum. And that condition is chronic alcohol abuse. So although I do not know these individuals from whom these brains were obtained, nor do I know their medical history, the appearance of the cerebellum in this specimen is highly indicative of chronic alcoholism compared to what we see in a healthy-looking cerebellum. And this alcohol abuse that I suspect might have afflicted this particular brain is likely to have impaired the coordination of gait and other aspects of locomotion in this individual in life.